You can ask questions of clarification and challenge him a bit. We have 2 announcements. Katie, do you want to go first? Or Mr. D, do you want to go first?
Katie, go ahead.
Okay, so I'm working with Andrew. Should I stand?
Sure.
On an IRT project, and we just wanted to give you guys a little 2-minute summary of what we're doing. So if you're interested, you can talk to us. So I'm sure a lot of people left over know someone with breast cancer, and some of you might even know that there are different types of breast cancer. One of them is called ER-positive breast cancer, and that's just for people who have the estrogen receptor. So that's the type of cancer we're working with. Why are we working with this? So there's a drug called tamoxifen that's commonly prescribed for ER-positive breast cancer patients. But unfortunately, like a lot of other drugs, people can develop resistance over time. So we're looking into the cell pathway that promotes tamoxifen resistance. So we're doing this by mutating a protein called 4-ABP1. We're mutating it in the hopes that our mutation will cause it to bind to another protein called EIF4E with greater affinity. Well, why does that matter? So if EIF4E doesn't bind to 4-ABP1 and it's loose, it can lead to the expression of oncogenic factors that promote the tamoxifen resistance. So we're hoping that our mutation will decrease the production of these factors that promote the drug resistance. So the first thing we're going to do is mutate the 4-ABP1, and then we're going to grow it in E. coli, isolate it, purify it, and then sequence it to determine its binding affinity. We'll keep you posted. Thank you.
Before you go away, come on in. There's plenty of seats available.
You're going to sit in front to
My announcement is just a very quick one. There's people interested in IRT Journal Club's Project 80 right now. All the groups are full, and a lot of you guys are wondering what more you can do right now. If you're attending Journal Club, if you're asking questions, and if you're trying to read some of these papers beforehand, then you're doing all you can right now. So we will look at picking up more people for all those groups in around, most likely December, but we'll keep everybody posted here. Yes?
Project 80 proposals are due by the end of the week, by the way, if you're involved in that.
Okay. Arjun!
So, hi guys, if you don't know me, my name is Arjun Patel, and my paper is the molecular basis for nicotine as a gateway drug, or in layman's terms, seeing whether exposure to nicotine causes a greater likelihood of addiction to other agents or other drugs. Some terms to know before we get started. Gateway drug. You guys have probably heard the term gateway drug in terms of like marijuana, but in this paper they focus on nicotine and as a gateway drug of cocaine. Priming, when exposure to one stimulus influences the response to another stimulus. So again, nicotine, cocaine. Phosphate and delta phosphate. All you need to know is that phosphate is a gene that codes for delta phosphate, and downstream delta phosphate causes an addictive behavior.
What does downstream mean?
Downstream meaning like when phosphate is expressed, it will eventually cause addictive behavior. It might act as a receptor or transcribe other genes that will cause addictive behavior. And you should know that accumulation of phosphate causes addictive behavior. So the scientists that did this research based their studies off of this epidemiological study from 2012, where they asked United States adults 18 to 34 years old who had ever used cocaine when they started smoking cigarettes. And a whopping 87.9% of them said that they used cigarettes before cocaine. Right now, as of this pie chart, you can't really tell that there's a causation. Correlation does not imply causation, so that's what the scientists wanted to research about. So they did that in 3 ways, 2 physiological markers and 1 molecular marker. The first physiological marker was locomotor sensitization. And that's an intimidating word, but it basically means how active the mice are. So when they're more active, it means they exhibit more symptoms of addiction. So it basically means when repeated administration of a stimulus leads to the amplification of response. So repeated administration of nicotine leads to addiction to cocaine and more activity in the mice. So they measured it in total distance traveled by the mouse, So, you know, the mouse would be in its cage and they take a video and then they basically measure how far the mouse traveled. So, the first chart. The things I want to focus on are what I outlined in the red boxes. And that's basically 24 hours of priming. So they give the mice 24 hours of nicotine. They expose it to nicotine, or they put nicotine in the water bottle of the mice, and then after 24 hours they give it cocaine. Or they give it water for 24 hours, pure water, and then cocaine, or nicotine and then cocaine. After 24 hours, you can see there's no real difference between their locomotion and their activity. And that's because, as we'll find out later, uptake of nicotine has to be done for a long time. It can't just be spontaneous. So that's what chart B shows. Locomotor sensitization after 7 days of priming. So the mice were given either 7 days of water, 7 days of nicotine, and then cocaine. And you can see that after day 11, which is the last day, nicotine, the mice that were primed with nicotine and then given cocaine had much greater locomotion or activity. Questions?
Did they have a baseline, did they mention this in the paper, before they added drugs of their just normal baseline of hyperactivity or regular?
They used a control that was, I believe it was water desaline.
But, like, were they all from, like, the same, like, genetic pool? Because maybe one was, like, more hyperactively than the other, and that could have varied the results.
I would assume that they didn't mention it, but I would assume that they did.
Okay.
I think they probably did.
Yeah.
They would be on the.
They generally, when you order these knives, they're all basically genetically identical.
Chase?
How does the mice's movement correlate to their addiction to cocaine?
Okay, well, they didn't mention it in the paper, but basically, I'm not really sure actually. So. Then the second one was a molecular marker, phosphate expression. Gene that when expressed causes addictive behavior, like I said before, and that's an oversimplified version, but you should know that it's quantified through a real-time polymerase chain reaction. And it's a really complicated process, so I'm not going to go into that either. But if you guys know what PCR is, where they amplify DNA, they basically turned the mRNA from the neurons into cDNA, and then they did a bunch of tests or a bunch of reactions, and then they just measured how much the neurons phosphoresced. Forget that, because that's not really important. But phosphate expression, it's widely used in everyday life. If you listen to like a good song that you like, or if you eat good food, or the reason you have to check Facebook 10 times every hour, that's because of phosphate, or phosphate is involved. So again, 24-hour priming and then 7-day priming. So the mice were given water for 24 hours and then cocaine, or nicotine and then cocaine, and then the same thing for 7 days. And again, just like the previous graph, after 24 hours, the mice didn't really show much of a difference between water and nicotine priming. And actually, you can see that when the mice were given water and then cocaine, they had more phosphate expression. 7-day priming. I'll explain why I think they have more phosphate expressed in 24-hour priming in a second. But 7-day priming, again, is a previous chart that says that more motion, more locomotion is shown in the mice that were given nicotine and then cocaine, as opposed to water and then cocaine. Back to D, you can see that here the water to cocaine has more phosphate expression. So the mice that were given water and then cocaine have more phosphate expression. And they didn't mention the paper, but I would think that it's because because the priming needs multiple days to actually take effect. After 24 hours, it's not really going to make much of a difference. So if you're given water and then cocaine, there's a much greater difference in how much the difference of how addictive they are versus nicotine to cocaine. So water is not addictive at all, and cocaine is extremely addictive. So it's a greater jump than nicotine. Claire?
Was there statistical significance between the water to cocaine and nicotine to cocaine and D?
D? No, because it's 24 hours.
I mean, like when the numbers are hard for it to be considered statistical.
I don't know.
I wouldn't think so. Just like,
The error bars.
it doesn't say. It would say so.
Yeah.
So basically, what these graphs are showing is that they agree with the graph in the previous slide that showed that it takes time for it takes repeated nicotine exposure to result in this priming effect.
Yeah.
Hyperactivity. Hyperactivity.
Why use hyperactivity as a measure of addiction? Or, well, not in this graph, but in general, like before. Why use locomotion?
I don't know.
Okay.
They didn't mention it, so.
Julia?
I can get back to you on that.
Cocaine, locomotion?
That's it, right, because it's usually like, to get like the stop and
I mean, we use locomotion as a good test of a couple things. It's a good test of anxiety,
Yeah.
but that wouldn't quite make sense here.
Yeah, I knew that one, but I just don't know why.
It's also actually a very good check as to tolerance, but that doesn't quite make sense here.
Yeah.
So you'll see like a cocaine addict develops tolerance to cocaine in almost all behaviors except locomotion,
Okay.
which is kind of weird.
The phosphate expression, of course, is just the direct measure of.
Yeah. This is probably the most reliable one because it's in the neurons, in the nucleus accumbens, which is involved in the reward pathway and addiction. And then the third marker was conditioned place preference. If you remember, Julia presented a few weeks ago on only conditioned place preference. So it is a reliable way of measuring addiction, and that's what they did here. So it's used to measure motivational effects of drugs. So how long the mouse stays where it's administered cocaine. So on the next slide, I'll explain that a little bit more. But silly example, if you will, it's not addiction, but in Tom and Jerry, when Jerry is like chased by Tom, he'll go into his little mouse hole. He's conditioned to know that he's safe in that place. And if you ignore the graph for a second and just look down here, you can see that basically this is also a simplification, but the mouse is in the cage and it's administered cocaine in one half of the cage, and they measured how much longer it stayed in that one half of the cage than the other half of the cage. So you can see if you go up to the graph that the mice that were given water and then cocaine stayed in where they administered cocaine for a shorter amount of time than the mice that were primed with nicotine. Does that make sense?
How long did they track the where the mouse went? Like for what period of time?
I think it was an hour. I'm not sure. Julie?
How long is the conditioning?
Well, this is the 11th day. So 7 days of conditioning, and then they gave them the cooking. So, summary. All data agrees that after 7 days of priming, after an extended amount of priming, the nicotine primed mice are more likely to become addicted. Not necessarily, not definitively, but they're more likely to become addicted to cocaine. And therefore, the scientists concluded that nicotine does act as a gateway drug, and it does have a priming effect on cocaine. I'm not done, sorry. Okay, so, sorry about that. So, this is basically explaining the molecular pathway that, how cocaine causes addictive behavior. there's a enzyme called HDAC, which I'm going to explain what that does in a second. But in organism that does not have HDAC, which is this top part of the picture, cocaine causes acetylated histones. Histones, if you guys don't know, they're proteins that DNA wraps around so that it can be stored more efficiently in the nucleus of the cell. Because you have 6 feet of DNA approximately in each of your cells, so it needs to be organized. When a histone is acetylated, it unravels slightly so that the portion of DNA can be transcribed or copied to make proteins or more DNA. So cocaine causes acetylated histones, which causes phosphatidylcholine in this case, which causes addictive behavior. In the human body, there's this enzyme, like I said, HDAC, or histone deacetylase, which deacetylizes the histones, therefore inhibiting that entire pathway, or inhibiting that part. So that therefore causes less addictive behavior. But what the scientists found is they actually came up with this model, this last part of it, which explains that nicotine inhibits the inhibitor of acetylates. So if something inhibits the inhibitor, then it allows for the pathway. So it allows for this part of the pathway to go on. So addictive behavior.
Do you know if HDAC only targets the histones related to phosphatidylcholine encoding, or just sort of?
I think they target any histones that have acetyl groups. Yeah, there's more specific versions that probably are involved, but this is.
When you talk about the phosphatidylcholine and then leads to addictive behavior, is that because it triggers different hormones to be secreted and different, like how does that work? What is the significance of it coding, right?
Okay, so phosphatidylcholine, meaning it makes delta phosphate. Sorry if that's not clear. It makes, so phosphate is transcribed and then translated to make delta phosphate, which causes. Does that make sense?
Do you know how it causes addictive behavior?
No, but they talked about how, not specifically now. I can get back to you.
What would happen, I don't know if they mentioned this at all, but what would happen if they were addicted to cocaine and then stopped using cocaine? Nicotine. If they were addicted to nicotine and then stopped using it and then started using cocaine, would they have the same effect of more addictive behavior?
For a certain amount of time, but after a certain amount of time, the effects of nicotine inhibiting HDAC would wear off. Does that make sense?
Well, if they stopped using the nicotine and then started using.
Yeah, so that would be what would happen here if the regular HDAC.
Okay.
So this nicotine priming of addictive behavior, would that go away once all of it left your system?
Yeah. Wait, could you say that again?
I don't think so.
If all of the nicotine left your system, would that stop the priming behavior? Would it stop inhibiting HDAC?
If all the nicotine is gone from your body?
Are you sure?
I mean, if there's no nicotine to inhibit the HDAC.
I think the point is that HDAC is epigenetic, so that the effect is long term.
In the paper, but I'll do.
It's probably just because I was priming and not using cocaine. After the probably more lasting changes. But just like it might take black linger for a little longer than after the nicotine leaves your system. But I wouldn't expect very just from what I've seen.
Yeah, so maybe epigenetically, but yeah, that makes sense.
You can tell me I'm wrong.
No, I mean, you know bad addictions. Okay, so most importantly, why should you care? Popularization of e-cigarettes, that's what the scientists focused on in the paper. They emphasize that a lot because as opposed to traditional cigarettes, e-cigarettes don't have any anything diluting them, any toxins or anything diluting the nicotine. So you're receiving pure nicotine, water vapor, and some flavorings sometimes.
So would this actually prime your, like e-cigarettes would actually prime your brain, I guess, more for like addictive tendencies for cocaine?
Mm-hmm. So yeah, that's what they were saying.
Would e-cigarettes prime you more than regular cigarettes because they don't have these toxins and dilutes?
Well, yeah, and there's nothing to dilute, so it's basically you're getting pure.
Does phosphine just affect the cocaine pathway, or is it?
It's involved in a lot of pathways. It's a widely used protein. Other questions? No? Okay. Proves that there's validity in the gateway drug theory. I hear a lot of the time some people don't like to believe in the gateway drug theory, usually when they're talking about marijuana, but that's something they could test further. But it proves that there is some validity. Now that marijuana is being legalized, that's something it's being legalized more and more. That's something they could test THC, which is the main component of what makes you high in marijuana. So they could test that, and alcohol, another legal drug. Thank you, Mr. D, Claire, Andrew, Jackson, Sydney, Mark, Catherine, Amanda, and Jake for preparing.
Any questions?
You go back like a couple steps. Yeah, how does cocaine, does cocaine specifically acetylate histones? So
No, it activates these. If you can see, it's small here, but it activates this pathway where these acetylate things. So indirectly, but I simplified it so it'd be easier to understand.
With the nicotine, like if you were to take it for a longer time, you eventually like the effects weren't as high. Would that be more to cocaine than like if you just did it for a day?
Yeah, that's actually what that's the point they were making in these in two of these graphs. Sorry, that if you're only doing it for 24 hours, which is not a significant amount of time, the nicotine will not work to prime addiction to cocaine.
So you said that like they condition for 7 days and then they test out London. Why is there that gap?
It was so if I just go back, so day one, these are they're the same mice, I believe. So they had day one, so they were conditioned for 24 hours, and they're given cocaine for 24 hours, and then conditioned for 7 days, and then 3 days after that they tested.
So the high bars here, they would indicate
that it's increasing.
It would increase phos-B, which would mean more pleasure.
Well, no, locomotor.
Well, but in the other one that did phos-B, right? You see the same thing. So more pleasure and more locomotion. So the effect of cocaine is greater.
So you're more sensitive.
But they weren't continuously administering the cocaine to them, right? Like are you saying that? Sorry, I'm just confused. So by day 11, were they still getting cocaine every day? Yes. Okay.
So like, essentially, was the nicotine making the cocaine even more addictive than it would be?
Yeah. So going back to this slide. So this B part is HDAC activity. So that's what you have normally. So if you were to take cocaine, there's the enzyme histone deacetylase, which removes the histone or removes the acetyl group from the histone. So you can't transcribe phos-B, which leads to addiction. And nicotine inhibits the enzyme.
So did you say HDAC inhibits the thing that cocaine binds to?
No, cocaine causes the acetylation of the histone, and then HGAC removes that acetyl group. So they're two separate, but they
So
do.
cocaine happens and then HGAC takes it away after.
Okay, I don't know if this is in the journal, but for just kind of, I mean, we're talking about HDAC, that inhibition, I guess, leading to addictive behavior, really. Would that also apply to other kind of addictive substances, or?
That's something they could test further. They didn't mention it in the paper, but seeing as cocaine isn't like there are different families of drugs, so there's like cannabinoids and cocaine is a different one, and then alcohol obviously. So that's something they could test further, but they didn't.